So we're going to start this last session with our keynote talk from Ava Amini from Microsoft Research on a little bit different topic, biophysics and protein design. So it'll be really nice to see her talk.
Thank you all so much for being here and for the invitation. Hopefully we can keep the energy high and enthusiasm high for this last session of the day. And it's my pleasure to present some of the recent work in our group in biomedical machine learning in Microsoft Research based in Boston, specifically focusing on our efforts at the intersection of generative AI and protein design. So to ground ourselves first, I'd like to take you down to the world of the nanoscale, where here what I'm showing you in this image is an illustration from scientist and artist David Goodsell. And what you're visualizing here is the boundary of a nuclear membrane in a cell at 1 million times magnification. And I think oftentimes as computational biologists or computer scientists, we maybe lose sight of what we are talking about when we think about the world of individual molecules and how they interact and function together in concert in a cell. And so what you're seeing here, as you may have guessed, is that these individual molecules that you see colored are proteins, right? As we all know, biology is actuators. And so our real question that's motivated a lot of work in our group is this question of generative design, specifically asking whether we can harness the power of computation to design new proteins to unlock new functional or therapeutic capabilities. This generative ability would unlock new opportunities across industries, applications from basic biology all the way to ultimate therapeutic implementation. And so to highlight kind of what we think about when we think of these applications around protein design, 3 examples that I'm showcasing here. The first is the creation and engineering of new molecular tools, such as gene editors or base editors that can really transform and advance synthetic and molecular biology, to the design of new enzymes that can catalyze new chemical reactions that are unseen before in nature. And ultimately, where a lot of my interest lies is in the design of new therapeutic agents, biologics or even therapeutic enzymes that could ultimately be deployed therapeutically, such as the enzyme asparaginase, which with some clever protein engineering has actually been clinically approved as a treatment in certain leukemias. And so really our first question is, okay, if we are going to set off on this task of trying to build AI to understand and design proteins, we have to think about how we represent a protein and what scale or what modality we want to tackle this problem at. And of course, right, in this community, there's been tremendous work across individual pillars of this pipeline, right, from protein language models such as the ESM family or the CARP family, to structure generation models like RF diffusion and others, as well as sequence to structure prediction models like RosettaFold or AlphaFold. And ultimately, where we in our group like to think about this problem is, can we integrate across modalities and leverage principles of biological grounding to build new AI models for protein generation in a multimodal fashion? And so today I'm going to focus on our efforts towards this end, speaking a little bit about some work that we've done in biophysically informed methods for structure generation, but really spending the majority of the talk and the majority of our focus on sequence generative models. And so thinking first about structure, what we were really motivated and inspired by was to think about how proteins naturally and biophysically fold to adopt a final structure. And as you may know, as a protein is being made and translated in the cell, initially it can adopt kind of this random unfolded configuration. And during that folding process, you have the sampling of different conformations to arrive at a final 3D configuration that's going to be energetically favorable. And what's really beautiful about protein structure is that we can very precisely and perfectly define the backbone structure according to these bond angles between the atoms that comprise the protein backbone chain. And if we think about folding as a high level of abstraction, you can think of it as this analog that's connecting these unfolded states corresponding to a random or pseudo-random set of angles, and a folded configuration where we have an inherent structure to the set of angles that define the ultimate 3D geometry and shape of a protein's backbone. And so looking at this process, a student from James Zhou's labs, who interned with us previously, Kevin Wu, had the concept of thinking about this native biophysical process of protein folding and using it as inspiration to define a new generative model for protein backbone design. And the concept here was, if we can define this process of going from an unfolded state to a structured and folded state as a stepwise denoising, if you will, that we could perhaps pose this as a diffusion problem to now build a generative diffusion model to do exactly this unnoising process framed as a design one. And so that is exactly what Kevin spearheaded and that we implemented in a model that we call folding diff or folding diffusion, where you can see some samples from generation stepwise from this model here on the right that effectively learns to unnoise from a random configuration of angles to learn the patterns of a structured state. And really the backbone to our method is a powerful class of generative models that are known as diffusion models that have been introduced previously in a couple of the spotlight talks, where the basic concept is you have some data distribution. You're going to perform a deterministic corruption process stepwise to now progressively add noise to your data samples and use this to constitute a training set of examples from which you can learn a model to iteratively learn how to remove the noise by looking at these pairwise consecutive steps in this noising process. And so the concept is, right, you have this controlled corruption scheme, and you can then parameterize a neural network to learn how to iteratively remove noise from these examples to then be able to generate new samples at inference. And so with this backbone and methodological framework in mind, what we did was pose this problem of structure design over these sets of protein backbone bond angles. And the concept here is, right, we define these angular sets. We can corrupt the data progressively over a fixed number of time steps until we get to a state of being indistinguishable from random noise. And then we can learn a neural network model in a generative fashion to be able to reverse this noising process such that we can generate realistic structures. And what I'd like to focus in on is kind of how the parametrization of this occurs with this angular representation. What's really beautiful and elegant about this framework is because we are defining our data set over the bond angles of the protein backbone, all the invariances and the geometries that we want to preserve about protein structure are inherently represented in that angular representation. So what this means is that the neural network that we learn as the denoiser can actually be quite simple, a vanilla transformer without any specific equivariance constraints or geometric constraints. So the key aspect of this formulation is kind of visualized here, where in folding diff, we've taken the equivariance requirements and baked that into the data representation itself, in contrast with some other approaches that have been proposed to now diffuse over 3D coordinates, where you have this diffuse point cloud of the atom coordinates in space, and then need to integrate these equivariance requirements into your neural network architecture itself. So I'm going to be very, for the purpose of time, really focus just a highlight of sampling of some of the empirical evaluations we did. We considered, okay, first of all, how well does our folding diff model actually recapitulate the natural distribution of these internal angular coordinates? Can we actually recapitulate structural motifs and key elements in protein structure that we would expect to see? And can we assess some of the generations for designability and diversity criteria? Now, I will say that, as you may know, right, this space of protein structure design is a very, very active one. And I think what was inspiring about working in this field and about this project in particular was we proposed an alternative formulation that has simplicity with respect to the efficiency and simplicity of the architecture, kind of inspired by the biophysical constraints of the problem and the actual biological question we're posing. So we really think that the open sourcing of our code, of our models, pre-trained weights and everything, really hopefully provides a building block for the ongoing work that has gone on to expand diffusion models for protein structure and now move to new classes of generative models as well. So so far, right, we've just given a taste of the work that we've done on structured design. But really, right, this is one part of the picture. And I would argue that ultimately, if we want to think about the general capabilities that we want for functional protein engineering, the real question we're trying to ask is, can we learn what amino acid sequences can give us a desired protein function? And so motivated by this question, our next step and our inspiration here was to ask, okay, what if we think about removing structure entirely and approach this design problem from the lens of sequence first? And pose the question of how can we achieve these principles of controllability by learning over protein sequence space? And really our underlying hypothesis was that sequence could constitute a universal design space for proteins. And the motivation behind this was that we have a tremendous scale of sequence data available. And furthermore, that when we want to engineer proteins, ultimately we realize them as their amino acid sequences. Whereas with a structural approach, there's this need for an indirect step to infer a sequence that would then fold and generate a particular structure. Furthermore, biologically, we know that many, many proteins fall outside this traditional structure-function paradigm, namely proteins that have intrinsically disordered regions that function very critically in a variety of cellular activities, and these traditionally have been inaccessible by structure design methods. And perhaps most importantly, from a data perspective, the number of sequences that we have available across data sets like UniRef greatly outnumbers, and by orders of magnitude, the number of structures that have been experimentally solved. And so this was really our inspiration and motivation to now think about building out classes of models that could learn from protein sequence data to be able to effectively generate new proteins well unconditionally, and ultimately give us a good foundation to be able to move towards this ability to prompt the model with different forms of conditioning to now be able to move towards this idea of functional design. And so the model that we have developed towards this end is a model that we call EvoDiff, which stands for evolutionary scale diffusion. And this is work that has been really spearheaded by an amazing applied scientist in our group, Sarah Alamdari, together with a really world-class team of complementary researchers and scientists at MSR. And the concept is, right, we want to be able to learn from this natural sequence diversity to be able to now generate new protein sequences that can hopefully execute desired functions. And like folding diff, the backbone modeling framework we've adopted is, again, a diffusion-based framework. But now we have a critical difference in that we're no longer in a continuous scheme of thinking about protein angular coordinates or 3D XYZ coordinates or other approaches. And really what we want to represent and learn over is this discretized representation of proteins as amino acid sequences. And the concept here is now, instead of having some noising scheme where we're adding Gaussian noise, we need to define a way to corrupt or effectively noise our source data to be able to train this diffusion style model. And what we adopt is a framework called the order agnostic discrete diffusion framework, where each step of the noising process is defined by the masking or corruption of these individual amino acid tokens in the sequence. And we perform this stepwise to be able to arrive at the final corrupted sequence, and as before, train a model to now learn how to undo this corruption iteratively. To train the EvoDiff model, we leverage a data set of on the order of 50 million unique protein sequences and a baseline architecture of a 640 million parameter CNN. Now, methodologically and from a machine learning perspective, what is really interesting about this discrete diffusion framework from a more theoretical grounding is that it actually provides a generalization of two other common classes of language models that are out there, namely mass language modeling and traditional left-to-right autoregressive language modeling. And so in this order-agnostic framework that EvoDiff adopts, we actually perform this corruption in every possible order, and each step of this iterative process is learned from a denoising perspective. This contrasts with traditional autoregressive models where you have a defined start token, you are tasked to predict the next token, and you perform this stepwise decoding in a single decoding order. While this gives us really strong performance in unconditional generation and actually learning the data distribution, the limitation, as we'll see later on, is that it's less flexible in terms of how you can try to condition on subsequences or parts of the input that you may want to hold fixed. And in contrast, right, mass language modeling, another common language modeling framework, amounts to learning the decoding over a single step of this objective, right? You have a fixed set of mass tokens, and you learn to predict the correct identities of those tokens at those mass positions. Inherently, this is not a generative task in and of itself, and it requires to actually sample from the trained model to now produce new sequences. And so diffusion gives this generalization over both these model classes, which lends distinct advantages. So in practice now, how do we actually generate new sequences at inference? With our trained model, we can now define a starting state of entirely corrupted tokens, and the model is effectively stepwise decoding and producing new amino acid tokens in this iterative fashion. And so in this visualization on the left, you see the outputs of our EvoDiff model stepwise to generate a new sequence from scratch. And on the right, what I'm showing you is a visualization purely post hoc in prediction of the resulting structure from that generation. And so importantly, what I hope you can appreciate is that this learning is entirely over sequence space, right? There's no notion of explicit structural information that's provided to the model. So with these two elements in hand, right, our discrete diffusion framework and this expansion over these evolutionary scale data sets, we wanted to focus our evaluations of the generations across these three axes, thinking about, okay, are these generations even biologically plausible? Are they designable? How diverse are they? How well do they cover the natural space of protein structure and function? And ultimately, what capabilities can we start to move towards from the perspective of conditional design? And so our first evaluations were really thinking about, okay, for an individual generated protein, how plausible are these? And so we adopted a framework where we think about generating a sequence, looking at its associated structure, and thinking about, okay, how plausible is that structure? And moreover, how consistent is that structure? Can it be realized by an amino acid sequence? And so in these plots, what I'm showing you is a pairwise correlation of the structural confidence of the predicted structure from samples from the independent test set against the sequence-wise self-consistency. And so ideally, you want to be in this lower right quadrant of this plot where you have high structural plausibility and confidence and consistency from a designability perspective. And what we can appreciate is that EvoDiff's generations kind of span this natural distribution fairly well. And if we look at some individual protein sequences generated by EvoDiff and look at their predicted structures, they're structurally sound and plausible across a range of amino acid lengths. So this evaluation is more on the level of individual generations. But now, what if we think about the overall distribution of samples that EvoDiff is producing? So thinking more now from a distributional lens, what we considered is how well our model could recapitulate the natural space of protein function and structure. And so in this analysis, what we did is we took test natural sequences, used an independent protein language model to generate embeddings for those sequences. And these embeddings have importantly been explicitly benchmarked to show separation across different functional classes of proteins that exist. And we can look at and compare the distribution across this embedding space for natural sequences compared to generations from EvoDiff, and compare these and visualize their overlap to see how well qualitatively EvoDiff is recapitulating this functional space. We can also try to measure this quantitatively by looking at a distance metric that measures the similarity across this embedding space. Ultimately, though, right, this plot is not super meaningful without understanding how other approaches or baselines are performing. And so that's exactly what we did next, comparing the generations from EvoDiff with samples from a traditional autoregressive model, which does slightly better in capturing this distribution unconditionally, but has less flexibility in terms of its power for conditioning. We can look at samples from a masked language model like ESM, where now we see a stark difference in the quality of the generations produced. And importantly, by the state-of-the-art sequences that are inverse sequence designed or inverse folded from a state-of-the-art structure generative model like RF diffusion. And here you can see that in a structural method, there's a strong bias towards a particular slice of this functional space. Now that may inspire the question of, okay, well, if this is over functional space, how well can these methods compare in their coverage of structural space? And again, this is exactly what we did next and performed a similar spirit of analysis, where now we're looking at an amino acid prediction of a secondary structure element and looking at the distributions of that analysis for generations from EvoDiff, as well as comparing to the test distribution. So first, if we compare now independent sets of natural sequences together, what you can appreciate is that there's this distribution over both helical and strand secondary structure elements. But if you'll notice in this bottom left corner of this plot on the right, there's this region that appears to be neither helical or neither strand in natural sequences that corresponds to regions that are intrinsically disordered and don't have an explicit secondary structure. And what is really exciting and encouraging to see is that with generations from EvoDiff, we recapitulate this same distribution of structural elements very, very well. And this is in contrast most starkly to structural generative methods that are quite biased towards preferentially generating structures that are biased for helical elements and missing this element of disordered space. And so the bottom line here is that by looking at these generations at a distributional level, that we can begin to understand how well our model is able to capture these elements of secondary structure that mirror those of natural proteins. So now, perhaps most excitingly, is, okay, now this gives us a framework and a basis for generating these unconditional samples, just generating samples from the model. To be able to move towards a paradigm to now think about functional protein engineering from a sequence-first perspective, we really need to think about our abilities to do conditional generation. And what's particularly beautiful about the discrete diffusion framework is that it gives us a very natural and inherent way to do some forms of conditioning inherently. And the concept here is that because we have done this discretized stepwise corruption and learned over all possible corruption orders, what that means is that at inference, we can fix some subsequences in a sequence and inpaint or generate the remainder of the sequence. What this amounts to in practice is masking those sequences that we want to generate over and decoding those portions, right? And that's, again, owing to the fact that this learning objective has learned over all possible decoding orders. And so in our work, we explored 3 different approaches to leverage this conditional generation capability. The first was to think about now informing a more stringent evolutionary constraint on the generation itself, where we could supply an alignment of multiple protein sequences of orthologs and now learn over this MSA rather than over a single protein sequence. And the concept here is that the entirety of the MSA, except for a single reference protein, a query reference, can serve as a form of conditioning, where now, given that context of the remainder of sequences in the alignment, we can generate a new query. We can also do kind of inpainting or scaffolding tasks, again leveraging this capability to mask these arbitrary subsequences in the overall sequence and be able to decode accordingly. And so I'll give you a taste over the next 10 minutes or so about how we evaluated and generated new protein sequences over these 3 types of application tasks, thinking first about this MSA-guided generation. So what is really beautiful about MSAs is that this is kind of a compressed representation of evolutionary information, where we can perform homology search to construct these alignments. And now the only difference as before is instead of giving a single protein sequence as input to the model, we can now leverage the entirety of the MSA and again perform this type of iterative masking task to now train a model that we call EvoDiff MSA, which is very similar in spirit, but now capable to operate over entire multiple sequence alignments of proteins. And again, we draw inspiration from this evolutionary perspective to curate a data set, to leverage a data set that spans MSAs across many different organisms and many different biological functions. What this capability to learn over an MSA in a single generative model gives you, and this is also in line with some of the work Sergey presented yesterday, is that traditionally, right, if there's a particular protein family that maybe you really care about, if you wanted to train a local model over that, you would have to curate that data set and learn the model to now be able to sample from it to generate hopefully new proteins of that family or class that you care about. But by leveraging the evolutionary information inherently encoded in an MSA as context, what this means is that we can effectively do an amortization of these local models over a single unifying model that's learned over across all these different MSAs. And to showcase what this kind of enables, learning over these multiple sequence alignments to now given the context of a set of aligned proteins, be able to generate a query sequence conditioned on that information, we see that we can actually improve the quality locally of these generations to now produce generated protein sequences with higher structural plausibility and better and lower self-consistency. And importantly, we're able to tread this balance between maintaining structural conservation where we care about it, meaning that we are able to match the constraints of the multiple sequence alignment provided as context, while still generating sequences that are novel with respect to the original information that was in that MSA. And so we think that this gives a flexible framework to be able to now condition on families of evolutionarily related proteins to balance this line between structural conservation and sequence novelty. So now thinking about, okay, right, the whole premise of this sequence is all you need hypothesis was that it would give us flexibility to do access regions of protein space that fall outside the traditional structure-first paradigm. And so what we were really excited about was exploring the possibility to now generate regions that could be contributing in intrinsically disordered proteins or intrinsically disordered regions. And again, to remind you why we had a hypothesis that we could achieve this, drawing back to these secondary structure analysis results, what we had observed was that in generations from our EvoDiff model, we were able to cover this secondary structure space, including this region of not inherently helical or not inherently strand-like elements. Meanwhile, structural approaches like RF diffusion are completely missing capitulation of this element, this region of secondary structure space. And so, motivated by this, what we set out to do was now to in-paint or generate intrinsically disordered regions within a protein of interest. And so the framework we set up was either using the single sequence EvoDiff model or given information and context from a multiple sequence alignment, could we mask out the region corresponding to an intrinsically disordered region within a protein, and then ask the model to, given the context of the surrounding regions and optionally the information from the MSA, generate or in-paint that region that we had masked out. And we can do some in silico analysis to now score that generated region for the likelihood that it is actually disordered. And so when we did this evaluation, what we were really excited to see was that across several examples, we were able to do this quite effectively in silico, as measured by these in silico evaluations of disordered regions. And so what you're seeing here on these bottom plots is now in green highlighted that masked portion that we masked out as conditioning to the model, corresponding to the disorder prediction score for the generation on the top versus the true intrinsically disordered region on the bottom. And what you can appreciate is that these sequence disorder scores correspond quite well across these examples. And so right now we're in the process of validating this approach experimentally to now look at some of these generations, particularly focused on intrinsically proteins with intrinsically disordered regions. So finally, right, the kind of a big paradigm in this realm of protein design is this notion of motif conditioning on a motif to now scaffold or generate a protein given that information as context. And biologically, this motivation is very grounded in the fact that protein function is often mediated through these interactions where there's a particular functional motif stabilized by the structure of the protein overall, and that motif is what's mediating the function or the interaction that the protein is is responsible for. And so we pose this counter-paradigm question of whether we could achieve success in scaffolding these canonical structural motifs using our model that has only learned from sequence information. And the short answer to that question is yes, that we can, specifically by this perspective of leveraging this subsequence context and masking as a form of conditioning. And so in this example where we can see that principle kind of come to life, what I'm showing you here is the structure of calmodulin, which is a calcium-binding protein responsible for calcium transport and signaling in the cell. And in green is that calcium coordinating region that is structurally and functionally responsible for that binding function. And here in the bottom is the amino acid sequence of calmodulin with the residues corresponding to that functional motif highlighted in green. What you can appreciate, right, is that these are discontinuous in sequence space. Even though structurally we have this continuity, they're discontinuous. So what we can do with EvoDiff is leverage these fragments, right, of these functional residues as a sort of biological prompt. And now, given this context of the motif, design a brand new protein sequence around that part, step by step from the bottom up. And so at the bottom is the visualization of the EvoDiff generation, and at the top is the predicted structure for the design resulting from this process. And again, at the top is purely a visualization, right? There's no structural information that's provided explicitly to the EvoDiff model. We were able to run this evaluation and analysis over a suite of different examples and benchmarks provided by RF diffusion. And what's really interesting is now when we compare the fractional success rate of generations from EvoDiff relative to RF diffusion, we see a comparable success rate in the terms of how many designs we can successfully scaffold. Digging a little further and deeper into this analysis, if we look at the relative success rates of the sequence-only model versus the MSA model, we see that they're not perfectly correlated across these scaffolding tasks that we evaluated on. And very interestingly, that if we compare success rates for RF diffusion versus EvoDiff, they're very divergent, suggesting that there could be complementary strengths comparing a structure-based model versus a sequence-based model. A couple of noticeable examples amounting to instances where the sequence-only model or RF diffusion has a relatively high success compared to an MSA model. In fact, this design has very few homologs that exist naturally. And from a novelty perspective, as perhaps you could expect, we can achieve higher novelties with a sequence-only model versus when we give that alignment information as context. And so from this perspective, what we're really excited about is that this is just one first step of a form of conditioning that we can leverage with this flexible diffusion framework. And ultimately, we see EvoDiff and this suite and class of models as a foundation to move towards this principle of controllable protein sequence generation, both from the lens of unconditional design, but also from across these types of in-painting or scaffolding types of tasks that are flexible across structural problems that we may care about, and also problems that fall outside this traditional structure-based design paradigm. So with that, I'll close by mentioning that our efforts in our group around protein engineering and protein design are just one piece of the broader effort across BioML at MSR. And all the code, model weights, data for both EvoDiff and FoldingDiff are open source and publicly available. And really what we see is the coalescing of efforts across these scales of biology from the level of individual molecules all the way up to the patient level such that we can build generalizable systems that can reason across these modalities to try to power relevant tasks in biological design and discovery. So with that, I'll conclude and really thank all of those in our group at MSR BioML based in New England, and happy to use the remaining time and for any questions and to talk further. Thank you.
Hi, thanks for your talk. I think, to me, beyond improved performance, the EvoDiff MSA unlocks completely new functionality. I think what protein engineers can use being conditional generation of variable sequence length by just introducing the knowledge and understanding of this gap token, which many of the sequence models don't have. Did you run any kind of ablation on in-painting gaps or diffusing two gaps?
Yeah, it's a great question. So this is actually something we're actively working on as we speak to give the flexibility across indels to account for that gap information. So hopefully stay tuned. So yeah.
Hi, great talk. I noticed that during like the functional scaffolding portion of it, there was still like, it seemed a fixed length of amino acids in the sequence. I was wondering if you guys were looking at maybe making that a little bit more dynamic, because some smaller peptides might be useful for some therapeutic applications or like manufacturing purposes.
Yeah, absolutely. So here in the evaluations over that scaffolding portion, you have to pre-specify that overall sequence length, right, for which you want to generate over. And what we did in this first pass of evaluations was, in the motif examples, conserve the residue-wise distance in between the discontinuous segments of the motif, and then, given a fixed overall length, experiment with different placements of that motif and sample over this possibility as well. So you're absolutely right that thinking about more clever ways and just evaluating over toggling that length in between the elements of the motif will give greater flexibility there. Yeah.
Thanks for the great talk. I'm wondering that like so the secondary structure or like this 3D structure can be also like the tokenized, right? And we can also do mask diffusion. Do you think that's useful, or like, yeah, I want to know your opinion about that.
Yeah, absolutely. So recently there has been a surge of this concept of tokenizing structure and then trying to even predict it, generate autoregressively according to that. I think, so the structural perspective is that you can never do as well with tokenization over structure than a pure, continuous or frame or angular representation that inherently represents structure natively, because of the fact that the tokenization is going to be some form of compression, right? So there is going to be some element of information loss. I think I'm eager to see what are the relative trade-offs. My intuition and hypothesis is that the approaches that ignore tokenization, that do not take a structured tokenization approach, will yield better results in terms of designability, consistency, et cetera, from a structural evaluation point of view. Now, there's a separate question of now, if you want to integrate across sequence and structure, what is the best way for doing that? Diffusion flow matching gives you a framework for thinking about cross-modal learning as well, and you can think about that in different ways. I think it remains to be seen. It's still an open question, but I have my opinion and happy to talk further on some of the things we're thinking about as well.
Okay, thank you very much.